Hi, I'm Jonathan Lakiro. I taught a synthetic biology lab course 2 semesters ago in fall of 19, and I'd like to tell you a little bit about that, some of the ups, some of the downs, and hopefully you can glean some info from me as you try and use synthetic biology in one of your classes. So I work at a small liberal arts university. We have about 1,200 to 1,000 students every semester. Our department actually does pretty well. We maintain between 200 and 300 students, mostly pre-med students. We have some pre-nurses as well. I teach a lot of the upper division biology courses, including immunology, virology, microbiology, genetics, and biochemistry. Those were a couple new ones for me. And I taught genetics for a while, and I taught the genetics lab for a while, and it was kind of disparate, a lot of random things. We tried to do a digital fruit fry lab, but they took that down, the one that I like to use, so I have to use a different one, and it just wasn't working well. I had students design their own primers, so I was trying to get them involved in sort of a cure-based idea, but what I ended up doing was learning about AR-CURE and trying to do synthetic biology. And so the number of students that I actually sent through it, I took students that were in the genetics course that I was teaching. So this is an upper division course. They're all juniors and seniors, the lab portion of that. And then I also sent my biochemistry students through there. It was my first semester teaching biochemistry. So I wasn't super familiar with exactly how labs should be run, but as I started digging, the only part of a biochemistry lab that we weren't really addressing was Westerns, which, as you may know, are notorious for not working anyway. Otherwise, almost all the aspects of the synthetic biology were really great for biochemistry students to actually engage in. So I replaced the biochemistry lab because I could kind of do whatever I wanted with the synthetic biology too. So all those students went through there. It was about 30-ish students in teams of 4. So when I actually tried to implement this, it was a full lab replacement. I did it as a complete replacement of our genetics and biochemistry labs. There were a couple things that we lost that we might typically cover in classical versions of those labs, but honestly, for the most part, students gained the exact same knowledge set. But they also got to have a lot more teamwork and got to work on experimental design. So my goal with all my classes, and especially the labs, learning some of the basic techniques that can be helpful, but a lot of my students are going to go on to be pre-meds, or they're going to go to grad school and work in a biology lab. And having come out of there 4 or 5 years ago, they're going to get trained in those areas anyway. Once they join a new lab, they might not know the techniques. They should know some of the basics. And so my goal is to get them to learn some of the basic techniques, pipetting, doing dilutions, but also to give them a story. How do you actually design an experiment? When they go to be interviewed about going to grad school, or what did they do as an undergrad? Why should we let you into our grad school? Well, I want them to have a story. Oh, we had this cool lab that we did where we got to design our own experiment and use different parts and piece them together and do ligations, and it just gives them a lot of cool stories to tell. So as far as my synthetic biology course went, the successes were what you could probably guess. A lot of great teamwork. Teams were formed and forged in the fire of trying to figure out and decipher exactly what they needed to do, which is exactly what I wanted. I told them very early on that I wasn't as interested in them creating successful machines. I was more interested in them effectively designing things to build, to work together to come up with a solution for a problem. The student interest in this was extreme. That might be a too extreme a word to use, but they're very interested in it. Our school, we don't have researchers. We're just all teaching professors. And for the most part, nobody else is really doing any basic research. And so to get them actually involved in using these kinds of tools, they were very excited for. It was very evident, even without me explaining, that these techniques would be very helpful. The students learned a lot. So that was a success. As far as actually creating parts that would work or machines that would work. We ran into a few problems. Some things that would help me. I'm actually going to do this next semester. So some things that I'm going to do that I think maybe you should think about. Some of the problems we ran into was a poor knowledge of technique. I was unaware of the lack of pipetting ability that some of my students had, the lack of dilution ability that some of my students had. I was kind of under the assumption that they would be able to deal with it. Some were. Some students took to it really well. Others needed a lot of hand-holding. And so that's something to think about to as you build your teams. I let them sort of choose their teams. But I think next time what I'll probably do is still have teams of 4, but have 2 advanced students and then 2 not as advanced students, and have them mentor each other with the requirements that they work together. Sometimes I do pull aside the more advanced students and say, like, hey, teaching someone something is one of the best ways to learn. And it gives you a story, and it gives me a story when I need to write a letter of recommendation for you, that I could count on you to actually uplift your team and bring them up to snuff, basically. But what I'm going to do next year is to actually have the first few weeks where we work on designing your experiment, how to actually use the website. What do you want to actually do? I'm going to have a big technique portion in there. Instead of approaching it as it comes, we're just going to have a couple wet labs where they just have to pipette. I'll probably do some formative assessments as well. Like, you have to pipette X amount of volume into this tube. You have to show me how to do a dilution. Yeah, things like that. We ran into a few problems with our reagents, which is on me. I have to do all my own prep. We supposedly have a lab manager, but they don't really do that. So did all my own prep, and I probably overheated my antibiotics for my plates. I've made 1 million plates in grad school, and this time I probably overheated them because my hubris got in the way. And it took too long to diagnose what was going wrong. We would get colonies to grow, but then it became clear that the colonies might not actually have the antibiotic in them. Some of them were reporting, some of them weren't. So we just ran into some issues there, and it just took a long time to diagnose. To be honest, it was a lack of available time on my part. We were having a baby at the time as well. I had a baby halfway through this lab. Well, I didn't, my wife did. But there was a lot going on. I tried to foresee some of these problems, but let's be honest, things just happen. So we finally got things diagnosed, but for the most part, it was a little too late to salvage a lot of the projects. But we did end up with some good results, as in students designed cool experiments. They got to do some wet lab stuff. Even if they weren't getting good bacterial growth, they still got to see what it's like to have something fail completely. Basically, as well, my students were supposed to operate pretty independently. I tried to be in the lab all the time. Near the end, I was forcing them to come and get me before they did anything. I had 2 TAs for this lab. That actually didn't help. My TAs for this lab were a little flakier than I thought they would be. They were extremely advanced students that had done basic science research and dilutions and knew how to pipette and done all kinds of stuff, but they actually didn't step up. And so I ended up having to deal with a lot more than I thought I would have to, which, as I said, I was super busy anyway. So I would do a lot of, okay, here's what needs to happen. Here's you're just going to use the kit's directions, because that's what you're going to have to do if you're in a grad school lab. Like, here's the kit, just use it. For the most part, students did okay. Silly mistakes. They would do tenfold more than they were supposed to. Instead of doing 200 microliters, they would do 2 mils, or they would, instead of 200 microliters, they'd use 20 microliters to elute, or silly things like that. For the most part, I didn't have reagent problems, other than sometimes they would leave things out, which for the most part really honestly didn't affect much. But Still was an issue. And so, yeah, those are the biggest problems that I saw showing up. Uh, Next time, I'm going to focus more on basic techniques. We're going to spend a lot of time doing that. I talked about that a little bit. I'm actually going to simplify the lab a little bit. This is not a separate course. This is a lab replacement. And so I was probably giving them a little bit more than they could chew. And so what I'm going to do is I'm going to simplify their lives. So I'm going to give them a few more pre-made parts. The idea of how to scaffold this that I'm probably going to run with is to give them a promoter and ribosome binding site that are already linked to just normal good ones, and then a terminator as well that we know works well, and then allow them to select a set number of reporters. I want them seeing something tangible. They took a bacteria that didn't glow green and now it glows green, or it didn't smell like bananas and now it smells like bananas, or whatever my reporter is going to be. So it gives them some choice. I want them to have some buy-in, but to have them link all the parts together, some of my advanced students got pretty close to completing a full machine, but a lot of them just linking two parts, mostly because the bacteria were just not cooperating. That didn't go well. Something else, make sure that if the facilities people accidentally cut the power off to your building, that they tell you and don't wait a whole week. So then when the minus 80 is back up to room temperature and all your bacteria are toast, that you know before that happens. That was great. I'm not super pumped about that. I did not have to go online. So I was teaching this class in the fall before coronavirus. So wasn't a big issue to move online. So I don't have experience with that. But for the most part, the students really appreciated the lab. They learned a lot, even though a lot of stuff just didn't work super great. But I just kept telling them, I was like, you know what? Honestly, welcome to grad school where things just don't always work. If you ever have any questions and want to just shoot some ideas at me, I will put my email up. You can look me up. I'm always willing to collaborate. But yeah, hopefully this was helpful. Thanks.